So, good morning for everybody. My name is Lucas Antonio Lisboa Ribeiro. I am a PhD student in the program of post-graduation of health science. I specifically work with Professor Leandro Barbosa, and my co-advisor is Dr. Gustavo Blanco. I work with the sodium pump, specifically in alpha 4 subunity. Uh, this is going to be my presentation for the class of bioactivity substances administrated by Professor Varotti. So basically, my article chosen is an article from Frontiers, Cells and Development Biology. It's an original research published in 2022. Is an A2 journal. And the article chosen it's called Genetic Ablation of the Sodium Pump Alpha 4 Results in Sperm Energetic Defects. So basically, this article is going to talk about what is alpha 4 and the importance for the sperm germ cells, right? The male germinative cells. So, and the work shows that if you delete the alpha 4 subunity of the sodium pump, it causes a defect in the sperm metabolism and it causes an infertilization. Okay, so basically giving you a nice introduction about the work. What sodium pump is. Sodium pump is a transmembranic enzyme that works with the fluid through cytoplasm to extracellular of the cell. And it works with sodium and potassium in the 3 2 molecules doing the gradient. So basically, it uses ATP for the energy and it trades the sodium and the potassium through the membrane. It's really important to this enzyme for a lot of functions in the organisms, principally in the mammals organism. Basically, sodium pump has two different subunities, the alpha one and the beta. The most studied subunity is the alpha because it's the part of it's the catalytic part of the enzyme. It basically has the part to connect ATP, the potassium, the sodium, and also in the extracellular camp, you have a place to connect other substances called cardotonic steroids that I'm not going to get in this now in this presentation, but basically are substances that can inhibit this enzyme if connected with it. So, alpha 4 is a type of subunity alpha that is exclusive founded in sperm germ cells, in male germ cells. Okay, so it's located basically in the flagellum of the sperm, and if it is inhibited, it can cause a lot of damage to the cell and cause infertilization. So basically, this work is going to show this importance of the alpha 4 subunity for the spermatozoa. Okay, so I'm not going to get in exactly all of the methodology they done, but I'm going to explain each one of the results after this part. So basically, in the start, they did a sperm preparation, which they get like from mice, from the Epididymis cauda. They get the spermatozoa and they did the swim up preparation, which is basically to you get out the sperm of the rat through a medium and use this for isolate the sodium pump. So here in this work, they use two type of animals. They use the animal that is control, which is basically called white type, and they use the KO mouse, which is a knockout mouse that don't have the alpha 4, so it don't produces alpha 4. Okay, they did determination of mitochondrial membrane potential and NAD and NADH determination, also to get like oxidative stresses. They did determination of ROS, which is reactive oxygen species, to see the damage caused of the rat that don't have alpha 4. They did also determination of lipid peroxidation, which you can see this in the membrane, right? The membrane of the cell. So if you have oxidation of this lipidium there, you have like a lot of damage caused. So they check this also. And they did a statistical analysis using T-student test, which is basically the best for this experiment, because they did the comparison in two groups: the white one, which is the control, and the knockout one, which don't have the alpha 4. And P was available significant when we're low to 0.05. So basically, when we talk about the results of this, they saw in the first step that the mice without the alpha 4 has different levels of glycolysis happening. How they measure this? They use the extracellular acidification rates. So they saw that the knockout mice displayed a significant reduction of this ACAR compared to the white type 1. So here in this graphic, it's easy to see. I just put in Portuguese here around the side the importance of this parameter they use, the ACAR, which is the extracellular acidification. Basically, what happens here is because when you have glycolysis, you have like a big amount of lactate showing up. So this acidificated, okay, this acidificated extracellular medium. So when you have like alpha 4 there, you can see here print in yellow. You can see that you have really high levels of acidification, extracellular acidification, showing that we have glycolysis happening in the right way. And for other side, you see here in the green one that you have low levels of glycolysis, not glycolysis, right? This parameter that is the ECAR, which is the extracellular acidification, but it shows that you don't have glycolysis exactly happening there. So this is the difference between the white and the knockout one. You see that glycolysis gets low, so it basically don't happen. So you have a problem of energy happening on the cells, the energy function happening on the cells. Also, they measure mitochondrial function, as I said before. They measure this function by OCR, which is oxygen consumption rate. Okay, so mitochondria basically consumes oxygen to bring the energetic demand. So it's necessary to have oxygen there. And in this graphic also you see in the yellow one is the white one, the control one. You see that you have a high level of oxygen consumption. And although when you see in the green one, the knockout one, rats don't have this consumption of oxygen. So this shows a low function of the mitochondrial side. This also shows the energetic side of the cells not working very well. We see here also the redox state of sperm is really altered. When you see like in these graphics, they show that the NAD and NADH ratio it's really higher in the knockout rats. So it has a significant difference between the control one and the knockout one, showing also that you have like situations of redox state not working very well as it was supposed to be in the control, like in the control one it happens. Um, Also, you see here in this graphic uh, that they found that the knockout one that don't have alpha 4, right? They don't have exactly an ATP consumption. So ATP is not really well consumed by the rats that don't have the alpha 4. You can see basically here this in yellow. So the yellow one, it don't have like a downing part of ATP. The blue one you see which is the control. You have like a consumption of ATP, and it's getting down because it's having consumption. So the in the yellow one you don't see these. It's all fluid, so you don't see the uses of ATP levels. And here also we have findings of the ROS and of the 4-HNE state here, which is basically when we are talking about this, we're talking about oxidative stresses. So they determine that the levels of ROS in sperm for alpha 4 and the knockout one mice was significantly higher than the control ones. So they saw the level of ROS reactive oxygen species. Yes, reactive oxygen species were higher when you have the knockout. So when you don't have alpha 4, you have higher damage of the system, of the cells, causing this big amount of ROS. And also they found a highly damaging product of the lipid peroxidation. So basically they analyzed this marker, the 4-HNE, which is a marker for showing oxidative stress in the lipid peroxidation. So also they did a blotting here showing right here in the B figure that in the white type you don't have this 4-HNE marker, but when you see in the control one you see the 4-HNE marker showing that this appears resulting in an oxidation of the lipidium from membrane, which is a problem. You have higher damage happening in the cells. So basically, as the title of this work shows, it was basically showing that the alpha 4, it's really important for energy consumption in the cells and for to be fertilizable, to the cells to be practical in do what they are supposed to do. Mostly when we're talking about energy utilization. So they saw that you have consequences in the energy production when you don't have the alpha 4 subunity of sodium pump. They saw reduction of glycolytic activity. Also, they saw problems in mitochondrial membrane potentials, and in the lipid peroxidation and oxidative stress that is really higher when you don't have this alpha 4, causing a lot of damage to the spermatozoa cells. So in conclusion, the results shows the importance link between the alpha 4 subunity of sodium pump when we are talking about energetics, using energy of the cell to work. So this causes completely infertilization in the cells. So basically that's it. Thank you so much.